Hello everyone, welcome to Ritika's online teaching academy. Today I will talk about the C3 cycle. Um, as we have studied in the previous lecture about the light reactions, chemiosmotic hypothesis, so we know that the photosynthesis has two types of reactions. The first one is the light reaction, the another one is the dark reaction. So today I will talk about the dark reaction. So this dark reaction is also known as the biosynthetic phase of photosynthesis. So now I am sharing my screen with you. Okay. So this biosynthetic phase of photosynthesis involves the chemical Calvin cycle, which is also known as the C3 cycle or a dark reaction. So this biosynthetic phase of photosynthesis does not directly depend on the light, and it depends upon the products of the light reaction, which are ATP and NADPH, and the other things which it requires is carbon dioxide and water. This phase of photosynthesis is also known as light independent phase or a dark reaction. Dark reaction name is given by the scientist only to differentiate it from the light reaction. But dark reaction is a misnomer. Calling it as a dark reaction is a misnomer. Why? Because uh, we cannot say that it occurs in dark or in night only, because it also occurs in the presence of light. So we can say that dark reaction of photosynthesis are not of light, as they utilize the products of the light reaction. Now next thing is how these ATP and NADPH are used in dark reaction or biosynthetic phase. We have seen that this biosynthetic phase or Calvin cycle uses ATP and NADPH, which are energy rich compounds and which are formed in the light reaction. So how they are going to be utilized for the synthesis of a carbohydrate molecule. So for that, many scientists have done experiments in late 1940s. Then in 1951, scientist known as Melvin Kelvin experimented on green algae. What he did, he subjected the photosynthetic organisms, which are green algae, with the radioactive 14 carbon dioxide for a few seconds. Then with the help of chromatographic methods, he searched for the metabolites in order to know whether any metabolite has incorporated that 14 carbon in it. So in this way, he discovered how carbon dioxide assimilation occurs in photosynthetic organisms. So the first compound which he got was the 3-phosphoglycerate, which is abbreviated as PGA. So that's why we call this reaction, dark reaction, as a C3 cycle also. So what are the different names for this dark reaction? First one is the light independent phase, we can also say to it Calvin cycle as well as C3 cycle. And because it is a biosynthetic phase, so we can say that dark reaction is a biosynthetic phase of photosynthesis. Okay, so these experiments now confirm that 3-phosphoglycerate or PGA is an early intermediate in dark reaction. That's why it is named as a C3 cycle. Okay, so the Kelvin cycle, as the name indicates, it is a cycle, means it occurs in a cyclic manner. So how many stages are involved in Kelvin cycle? There are mainly three phases. The first one is the carboxylation. So as the name indicates, carboxylation, here carbon dioxide is used for the carboxylation of RuBP. What is RuBP stands for? RuBP is ribulose 1,5-bis phosphate, a 5-carbon acceptor. and it, when accepts one molecular carbon dioxide, it forms two molecules of 3-phosphoglycerate. Okay, now scientists thought of that, how a 5 carbon acceptor, which is ribulose 1,5 bisphosphate, when it accepts carbon dioxide, it should form a 6 carbon compound. Instead of that, it is forming two molecules of 3-phosphoglycerate. Okay, so means that this molecule, after accepting the carbon dioxide, it cleaved into the two molecules of 3-phosphoglycerate, and the enzyme which is used or which catalyzes this reaction is ribosco, which stands for ribulose bisphosphate carboxylase oxygenase. In this reaction, only the carboxylase activity of this enzyme is used. Oxygenase activity of this enzyme we will discuss in later lectures. Okay, so the second stage is the reduction. Reduction occurs with the help of a NADPH. As we know that in reduction, hydrogen molecule gets added to another compound or it reduces another compound. So we know that the first intermediate is 3-phosphoglycerate. So this NADPH reduces 3-phosphoglycerate into triose phosphates. This triose phosphate with a series of reaction will lead to the synthesis of sugars. It means that sugar synthesis is not a simple process. It also involves various steps or a series of synthetic steps. The third one is regeneration. Regeneration, as the name indicates, molecule again appears or again regenerate. So which molecule regenerate? Here it is RuBP, ribulose 1,5 bisphosphate. Why it is regenerated? In order to continue the cycle again. If the cycle continues uninterrupted, this RuBP is regenerated again and again in order to continue the cycle so many times. Okay, so now let us understand all these three stages in the form of a cycle. So first is ribulose 1,5 bisphosphate, which accepts carbon dioxide and water from the atmosphere and forms the first intermediate, which is 3-phosphoglycerate. Second step is reduction. This step involves ATP as well as NADPH. ATP is for the phosphorylation of the 3-phosphoglycerate, while NADPH is used for the reduction of 3-phosphoglycerate. So here hydrogen of the NADPH is used for reducing 3-phosphoglycerate. Okay, so ATP gets converted into ADP plus inorganic phosphate, and NADPH gets converted into NADP plus. Okay, so what is the product of this reduction process, which is triose phosphate. Now triose phosphate will then lead to the synthesis of sugars and starch, which is again a complex series of synthetic steps. Now this triose phosphate, third step is it regenerates the ribulose 1,5 bisphosphate. So how this regeneration takes place? This regeneration also occurs at the expense of one ATP molecule. Here ATP gets converted into ADP and inorganic phosphate. Again it does the function of phosphorylation and forms the ribulose 1,5 bisphosphate. So this ribulose 1,5 bisphosphate gets again free for the continuation of the another cycle. Now in the next slide we will understand that how many carbon dioxide, water molecules, ATP and NADPH are used in a Calvin cycle in order to synthesize one glucose molecule. Okay. So in the carboxylation step, one carbon dioxide and two water molecules are used per cycle. Per cycle means what? In each cycle, one CO2 molecule is fixed and two water molecules are captured from the atmosphere. The another thing, two molecules of 3-phosphoglycerate are synthesized by the reaction of ribulose 1,5 bisphosphate with carbon dioxide, one molecule of carbon dioxide. Now reduction step, it involves two molecules of ATP for phosphorylation per cycle. Per cycle means in each cycle it requires two ATP molecules and two molecules of NADPH for reduction of per CO2 molecule fixed. While regeneration step, it requires one ATP molecule where ATP molecule phosphorylates and regenerate RuBP in each cycle. So we can say that in one cycle of Kelvin cycle or C3 cycle, here one carbon dioxide, two water molecules, three ATP molecules and two molecules of NADPH are required. So this is the case of the one cycle, but in this one cycle only carbohydrate or sugars are not going to synthesize. So how many tons of the cycle are required in order to synthesize one glucose molecule? That we can easily understand by the reaction equation of the photosynthesis. This reaction equation of the photosynthesis shows that 6 CO2 molecules plus 6 water molecules are used, and they in the presence of sunlight and chlorophyll form one molecule of glucose, which is 6C6H12O6 plus 6O2. So 6 CO2, and we have seen that in one cycle 1 CO2 is used. So for adding 6 CO2 6 cycles are required. And how many molecules of water? We can say that for hydrogen, in the hydrogen, how many hydrogen molecules? 12, which are coming here in glucose as H12. So how many water molecules in each cycle? We can say two molecules of water are used in each cycle plus 6 O2. This oxygen is coming from the water molecule. So this oxygen is not coming from the carbon dioxide. We get it from the water molecule. So we can understand the whole thing in a form of this table. Requirements for the generation of one glucose molecule. Total carbon dioxide which are required are 6 molecules. ATP for 6 tons of the cycle, which are 3 into 6, which is 18. NADPH 2 for each cycle. So for the 6 cycles, it will require 6 into 2, that is 12 molecules. So these many molecules are required for the synthesis of one glucose molecule, and it takes place by 6 tons of the cycle. Means when 6 tons of the Kelvin cycle is completed, one glucose molecule is synthesized. So for the formation of one glucose molecule, 6 tons of the cycle are required. So in the NEET exam, it may be asked that for how many ATPs are required for each cycle, for each Kelvin cycle, or in each this biosynthetic phase or dark reaction, how many total ATPs are required. So for each cycle you can say 3 ATP. But for the synthesis of one molecule, we can say that 18 molecules of ATP are required. If the question is asked how many NADPH are required for synthesizing one glucose molecule, we can say that 12 molecules of NADPH are required. So these many questions are generally asked. We will discuss in the next lecture about the C4 cycle, and after that one questionnaire session of NEET exam. I will prepare a video for that. So please do like, share and subscribe my videos in order to be able to get more videos on in biology, which will be helpful for you to clear the NEET exam. Thank you.